All right, ready to go. So you click here to begin. And you get this info, you click to continue. And hopefully everybody got to this point. Now there is a little help screen over here. If you wanted to go in there for help, you can. But you can also get most of that help somewhere else, so I'll get rid of that. A little bit later, we'll probably do some sequence analysis here. So all the stuff that we work with, we can actually get the DNA sequence for it and put it in here, and then we could blast it and do stuff like that, but we won't do that right now. So we just have this thing. So we got our main window, which is called the data screen, and we got this thing, which is called our open and processed window. And whenever we open anything that we're going to use, it pops up in here. Up here, you've got all of these things. If you want to go get DNA, which we'll do, you have that. Some of these things are redundant, like this DNA button is the same as that DNA button. This little thing moves around. Sometimes it's at the bottom. Sometimes it's at the top. Oh, crap, went too far. And hopefully I can get it back. Come here. Uh-oh. I did not want to put it on the top. Come back. There it is. I can't get it to come back. Come back. Huh. Well, that was no good. All right, I'm just going to close it and open it back up and see what happens. But sometimes that thing hides. That's all I was trying to say. And you might have to go look for it. So I could just click back up here. Click to begin. Click to continue. Get rid of these things just so we have more room. All right. All right, so up here, there's the tutorials for everything we're going to do. Every time there's an assignment, you can first look at this tutorial to see how to do the technique. We're definitely going to do some microarrays. What else are we going to do? We might do a Western blot. We'll see. But you can just watch the videos for what you want to do. The one we're doing now is PCR digest DNA, and you click over there and you click on that and a video will show up. Hopefully you've watched that already. In here it tells you what all the different cases are. So there's a bunch of genetic disease cases. So if you want to go read about them, you would go read about them here. We'll definitely do one or both of these. I might do this one real quick today because this is somewhat relevant. You can actually do a little quick PCR diagnostics of the SARS virus. So this is like the first one. It's the same one we're dealing with now. Now we're dealing with COVID-19, but this is like MERS and SARS or those kinds of things. This one's cool. There's honeybee ecology. I don't know if we'll get a chance to do it, but it's pretty cool. But anyway, there's lots of these different things. And that's where you would go to read about your little case. But the one we're going to do now, I already know how to do it, so we'll just do it. So you go to DNA. So what the point of this one is, it's a cystic fibrosis disease, and there's a gene that is, let's go look at the case. So if we read about the case, and we go to cystic fibrosis, it tells us all about the case. And it tells you who the people are and all that kind of stuff. In this case, it's a little bit weird because we're not looking at the CF gene precisely. We're looking at this other locus, which just means a place on a chromosome. And this locus is tightly linked to CF. So, and we know that a particular version of this is linked to the mutant allele, and a particular version of this is linked to the wild type allele. It doesn't have anything to do with the gene, it's just tightly linked to it. Now, we haven't done much with linkage, and we will do stuff with linkage, but it just means that if you inherit the mutant CF gene, you inherit a particular version of this. So that's what we're actually looking at. So we have a point mutation in this thing that results in the loss of a restriction site. So if we're missing that restriction site, that means that it's that we have the mutant CF gene. If the restriction site is present, that means we have the wild type normal CF gene. Not going to read the rest of it, but you get the gist. And now I got to figure out how to get out of here without closing the whole program. See what happens. I think if I click this, I think I get out of it. Whew, that was good. All right, so what we're going to do is we're going to do PCR. So to do PCR, you have to get some DNA to use as a template. So we're going to go to get DNA. And then this window shows up. And we're going to go into a particular case. If we were doing a cancer case, we'd go into that folder. But we're going to do a genetic disease case. So we go into there. We're going to do a cystic fibrosis case. So we go into there. We're going to do case A. And we'll do the control DNA. And so I want you guys, so the assignment is for you to do the control DNA and then to pick one of these families and do the same analysis on them. And so the only thing that would be different is that you'd go in there and get the stuff from in there instead of in the control thing. So double click on that, and we always have these various files that represent the different things that we need. The .gen means this is genomic DNA. And genomic DNA means it's the DNA you would isolate from a patient, just total DNA. If we're going to do PCR, we'll need a primer. And then we're going to cut that PCR product with this restriction enzyme. And if it cuts, it's wild type. And if it doesn't cut, it's mutant. And then we can tell whether it cut or it didn't cut by running a gel. So we'll just go through those steps. So the first thing we got to do is get our DNA. And I'm going to hold down Shift, and that'll select all three of those. Sometimes a little click box shows up here, and you can click those. You know, Mac's a little bit different, but you just have to somehow click those. You can take them one at a time if you want, or you can just click and then hold down Shift and click again, and you get all three of them. Open them up, and they show up in this processed window thing. And so there's our DNA. But we want to do PCR. When you do PCR, you need a template, which we have. You need polymerase, nucleotides, and we just assume we have those. But we also need a primer. The primer is what makes it specific. And so we're going to click on this, primer. We're going to go to, this should actually say primers. That would be more accurate. And so if we go into here, we see this file here that contains some primers. And we click on that, and we open it. And then we've added this to our, think of this as like your benchtop. You have some DNA in a test tube there. You've got some primers. And we'll just assume we have the PCR mix somewhere. And this is the actual sequence of the primers. If we actually took these sequences and did a PCR, we would actually amplify up this gene. They're like the real primers. FP stands for forward primer. RP stands for reverse primer. Whenever you do PCR, the primers have to be pointing towards each other, and they'll amplify up whatever DNA is in between them. And so we have these things now. We want to do PCR. And I think what I've also done, the next thing we're going to do is get an enzyme. And so sometimes I've just gotten this enzyme ahead of time and put it in there, and that seems to screw things up. So it's better to do it just one step at a time. And we're going to do PCR on all three of these things. And this DNA says heterozygous. This is homozygous mutant. So these are just control DNAs. And these are homozygous normal. So these are the DNAs that we would use to compare to our patients to see if they were homozygous mutant, homozygous normal, or heterozygous. And the heterozygous ones are the carriers. So if we want to do PCR, and it's just kind of important to remember the order. You might just write down that what you do is, what you would do in the lab is you would isolate DNA, get some primers, do PCR, and then do a restriction digest. So we've got our DNA, we've got our primer, we're going to run PCR. So you go to run, we're going to run PCR, gives us a billion options, we just need this first one. And so now it generated PCR products that will contain DNA from that, whatever that gene was called, that's right next to the CF gene. So we've got that DNA in a test tube now. And now we want to cut that, and we want to run it on a gel. If we're going to cut it, we need an enzyme. Some of these cases will come with multiple enzymes, but this one only has one. So we're going to get that. We're going to open it. Now we've got an enzyme. So the next thing we're going to do is we're going to cut DNA, but we need to select the stuff that we want to cut. And we don't have to select the enzyme because it just knows that if you're doing a digest, this is like a restriction enzyme digest is what they call this. You would, it'll just do it automatically. So now we select, hold down shift, select all three of these, cut DNA. We only have one enzyme, so it'll cut it. And so now it's generated these things. It's like another 3 different test tubes. One of them has this PCR product cut with the enzyme, this PCR product cut with the enzyme, and this PCR product cut with the enzyme. And now we want to run these on a gel. All I've asked you to do in this assignment is to run these 3 things on a gel, but I'm also going to run these on a gel so we can see, we can kind of see what they look like compared. And let's see, it's a little bit hard. Let's see if I hold that down and shift, I can select these 3. The problem is if I come down here, if I hit control, it doesn't do it. And if I hit shift, let's see if I can select all this stuff. And then, that didn't work. I want to do this without using that enzyme. So I can't, so I'm not going to do that just now because that I can't figure out how to, I can do them separate. So first I'll just do this. So we're going to run a gel of our DNA PCR products that were cut with this enzyme. We could go through all these different steps. We could come up here and we could run a gel. We could run a gel and make it a DNA gel. Use the lab bench where you use a pipette and you do it all. But that gets annoying really quick. So we're going to use this thing, quick load and run. We're going to run a DNA gel. Is everybody still with me? Everybody got the microphone on? Okay, good. I just had this little panic moment that I was going to go through all this and none of you guys saw it. So now we got this DNA gel. And the quick way to do it is to just load the wells and run them in one step. So it just does it all for you. And we'll first do it. The video has us do this. Make these things barely visible. So you can see these things down here. But in reality, you wouldn't be able to see those. Sometimes you add the ethidium bromide right in the gel so you would see these immediately. Sometimes you run the gel and then stain it later. So this is like an example where we would run the gel and stain it later. These buttons down here let you stain it with different things. The one that looks most like what we usually see is, well, I guess this is officially what an ethidium bromide one looks like. And then this, I kind of like this view a little bit better, but any of these are okay. This is like what it would look like if you didn't stain it with anything. This is if you stain it with something blue, ethidium bromide, and I don't know, if you took a black and white picture, it looks like that. So this is our control DNA. We ran a PCR, and then we cut it with an enzyme. Down here, this says lane 1 is our heterozygous PCR product. Lane 2 is our homozygous mutant. Lane 3, they call it well or lane, is a homozygous normal. So if we look at 2, which is homozygous mutant, the mutant does not get cut with the enzyme. So this is just the size of the original PCR product that didn't get cut. The wild type, there's a restriction enzyme in the middle of that PCR product, and so it cuts it into 2 bands. Now we don't really know what size it is, but we can tell these are controls that just if you get 1 band, that means you just have the mutant allele. If you have 2 bands, you have wild type allele. If you have all 3, that means you're heterozygous. It means you have 1 mutant allele and 1 wild type allele. So even though there's 2 bands here, this just represents 1 wild type allele. So let's take a look at what it would look like if we didn't cut it. So we'll just run these PCR products out of gel. And I think they'll run on the same gel. Not 100% sure, but let's find out. So it ran them all together. So these PCR products are what it would look like before we cut it. So PCR just amplifies DNA up of a particular size, and then if you cut it, If it cuts, it stays the same size, or if it doesn't cut, it stays the same size. If it cuts, it gets smaller. If you got, so this is 2 mutants, this is 2 wild types, this is heterozygous. If I want to take a picture of that and put it in a report, I'd go to my snipping tool. Hope I got snipping tool down here. Hold on a second. I'd go to snipping tool, get a new little snipper thing, come over here, take my gel, cut it, and paste it. But what you can also do, so that this makes more sense, is you can it's better to like type in here what's in each lane. So what I would do is type what's in each lane and then do snipping tool to grab this whole thing so you have the labels in there. And you could do that in a couple ways. You could just say, you could say lane, you just type it in. Lane 1 is homozygous normal. You just type it in. But you can also come down here and say lane 2, you can say like, well, you can select this and then say copy into gel label, and it pops right in there. So that's a little bit easier. And then you can put it down here so you can read. You just drop it down there somewhere so you can read it. I don't know how all that stuff got in there. Oh no, that's all right, because this won't show up. So that's that. So that's what it looks like. So what we're doing, so what this is called, I'm just going to type this down here, because not because it's a label. I guess it could be part of the label if you wanted to. For some reason, it's not typed. Anyways, so what we did is we did PCR, and then we cut that PCR product with a restriction enzyme, and we got different fragment lengths because these things are different. If things are different, they're called polymorphisms in genetic terminology. And so what we did is we looked at restriction fragment length, because that's what these are. These are restriction fragments, and they're of different lengths. And they're different, so they're called polymorphism. We will cover this in more detail. The next thing we're going to cover in the lecture part is DNA variation, and that's why we're doing this one now. And polymorphism is a term for DNA variation. So this is called an RFLP. So just the different sizes is called RFLP. If you do PCR first, then it's PCR RFLP. So that's the name of the technique. You can do other things, like we could come in here and you can either right click on this or you can click set. And we could run this for a shorter period of time. I don't know why it didn't do that. It should run shorter. Maybe I have to hit enter or something. Maybe I have to hit set now. I just did that. All right, now run it. All right. So by running it for a shorter period of time, it didn't run as far. We can still see the bands, but it didn't run as far. Run it down you run it down farther, and they spread out more, and they run farther. Let's do one more thing, just to kind of show you what's going on. Oh, a good thing here is, let's say you keep doing this and it kind of gets messed up and you got a bunch of bands in here that you don't want. You can go up here and do clear. And you don't want to clear everything, because that'll get rid of everything to the left. So you just go down here and you just clear all the wells and everything, and you can kind of start over. But I just thought I'd just show you one thing. If we just run this DNA without doing PCR or anything, we'll just run it. We'll do this quick load and run business. So this is just the DNA that we started with. I don't know where this band came from. But notice how it's super huge, because this is where we loaded it, and it's running left to right. And the genomic DNA is really big because it hasn't been cut with anything, so it doesn't go very far. And then if we compare that to like a PCR product, The PCR product runs down here. I don't know what the heck that is. And so that's why we do PCR, because this is all of the DNA. And here we focused in on just one particular spot. So hopefully that's clear. And later on, when we do other techniques, you would just do other things. Like if you wanted to do, let's see, up here, if you wanted to do a microarray, you would click on chip. And you could do, there's two different kinds of microarrays. There's a SNP chip, and there's a expression one. This is the one that we've talked about a little bit in class. You pick one, and that's a whole different ball game, so I won't go over it. I just want to point out that there's different things. If we want to do a Western blot, we could, if we had a case, we could go to a case that had antibodies and get a Western blot, that sort of thing. All right. Any questions? Oh, let me see down here. I saw a little chat thing show up, but now I can't. It keeps going away. All right, let me move this out of the way. See if I can get this. The the genetic paper, the gene paper is due Monday, and the first assignment for this is due Wednesday. So a week from today. And then there's nothing else due next week. So I put the schedule on the course web, and you'll notice. So we're going to have quizzes, I think, every Monday, and those will just be short quizzes on Moodle. And then we're going to do several of these case it things. And at the end, we're going to have like our final exam is going to be kind of a slightly longer quiz, and then kind of a little project. Probably one of these. Probably some kind of case it project or something else, or maybe writing a little thing about CRISPR, or reading a paper or something like that. So so that's what we got going. The points, I think I put everything in the email, but the points worked out so that you get like 10 extra points just for the annoyance, just to keep the numbers straight. And so I also added in the 25 points to everybody's grade. It didn't really change your like midterm point grade thing, but it made it higher. So if you want to know exactly where you stand, just send me an email, and I'll send you all your points. No, unless you want to. Do you want to? My plan is to. No, that's a good question. My plan is to. Well, we might have class like this Monday, Wednesday, Friday, but but I consider them more like office hours, sort of. So I am going to record lectures for all the PowerPoints. We're moving on to chapter 4 next, which is genetic variation. So I'm going to just pick up there and go. So all the techniques you should have a handle on from watching videos, and that's all in chapter 3. And then so those you'll be able to watch whenever you want. So we call this like an asynchronous approach. So we don't have to all do everything at the same time. And then I'll be available for these kind of Google Meets. I guess my initial thinking is just to to have little discussion, like kind of run it more like a review session, sort of each day, where you guys have watched the video and then you ask questions. Um, let's start doing that Monday, Wednesday, Friday, and I'll record it in case you can't make it. And we can also pick other times if like this time's better for people. I don't care. I'm not going anywhere. I'm stuck in my house, so we can do it whenever. So maybe we can come to some kind of consensus as to whether we want to meet every Monday, Wednesday, Friday. That sounds a little excessive to me, um, but whatever you guys want to do. And you can also just do it on demand. If you want to schedule a little Google Meet time with me, you have some questions, just send me an email and set one up. And I don't know, can you guys set these things up too? Or do I have to set it up? You guys can probably set it up. Yeah, yeah, anytime. And I could always, or you can just ask me what you think a good time would be, that sort of thing. So this case it thing is pretty cool. Just remember the one thing that I've had to do quite a few times is clear this. And if you clear, I mean, you might want to clear everything. If I clear everything, it clears everything, and then we're back at the beginning. And that's sometimes that's what you want to do because it gets confusing and you've digested things twice, or you've got 8 different kinds of DNA and you don't know what's what. So you just clear everything and start over. Once you get the hang of it, it's super quick. Some of them are a little more complicated. The qPCR is pretty complicated, but I'm not even sure if we're going to get to that one. That's the honeybee one, and it's I don't know. It's a little much. So all right, let me. So any questions on the case it thing? I can't see the chat thing, so if you're chatting, just turn your microphone on and speak up. All right, does everybody feel comfortable? Yes. There's a video on CourseWebs that goes over this particular case, but that's the same video that you would get here. If you went down to this and then you went to this, that would show you the same video. And then there's also a thing called a walkthrough. So a walkthrough is like basically me doing this, but stopping and doing a screenshot and pasting it in as I go. So that's a walkthrough, and that just shows you how to do the technique, and then the assignment is like, okay, now go redo it yourself and give me, answer these questions. So there's an assignment on there called Case It One assignment or something like that. Other videos, so like this video, I won't be able to post it on CourseWeb. I think I can put it on Google Drive. So I'll probably make a folder for all videos on the drive and just give you guys access to that drive. Because the files are too big, so I can't put them all on the CourseWeb. But I can like put links to it on the CourseWeb or something. So I think tomorrow I will start going through the different chapters. So I would definitely. I wanted to point out. Let me see. I'm going to stop sharing this, and let's see. Can you guys see me? Okay. Just want to remind you of this particular thing here. This is our book. So some of you probably don't even have it, but that's what all the lectures are going to be coming from that. So go read the book. Particularly look at the figures. Genetic variation chapter can be a little bit. There's a lot of info in there, so we'll try to pare it down to the essentials. But keep an eye out for that. I'll get chapter 4 done tomorrow, and we'll move from there. No, nope. So we won't start any Google Meets till next week. So this we're still in transition week. Post the videos so that you can or post the PowerPoint lectures so that you can see them. But we won't start talking about them till Monday. And if you look at, I posted a new schedule on the course web, and so go look at that, and you'll know what's coming up. And so each week's kind of the same. So Monday, I can't remember which day's which, but like one day we don't have anything due, one day we have a quiz, one day a case of things due. So it's basically just lecture, quiz, and case it's each week until the end when there'll be a little bit bigger assignment. So it's it'll keep you busy, but it's you don't have to write any 10-page papers or anything. Um, we'll start doing that next week, and we'll see how it goes. So we'll definitely have them. We'll definitely be able to do Google Meets during those times. If nobody's showing up during those times, or if they don't work for people and we need a different time, we can reevaluate. But we'll start next week. We'll start on Monday at 11:45. we'll do that Monday, Wednesday, Friday next week. But I know some people's schedule might be all screwed up because of what's going on and who knows what you're doing. So, but let's just take it 1 week at a time. Yeah. No, you actually do have to. No, you don't have to run both the PCR uncut and cut, but you have to do the control DNA and you have to do one of the case studies. So you have to go in and do the Brown family or the Jones family or whatever the other family was. And so that's good. That's going to be the same. So all you're going to do there is you'll open a DNA file, go to case A. This is case A control. So I'm going to back up one level. If I can move this window, that'd be better. That doesn't matter. So let's say we want to do the Smith family. I'll take Carol, Fetus, and Ron, take that DNA, then get these primers, then run PCR, then get this enzyme and run a restriction digest, and then run a gel. And so that'll give you the genotypes of the parents and their fetus, which is why you would do this, right? So this is for genetic diagnosis. So I just figured that's better than doing just control DNA. That's kind of boring. And you may have to go like read, if you want to go read the case study to see which family you want to pick, you go to here, cystic fibrosis. And we got case A, we got case B is the, I don't know who these people are. I don't even know if it gives you their last names. How the heck are you supposed to know which one's which? Carl and Maggie. Oh, this is in case B. So case A is all the same. You just, oh, here we go. Sharon Brown. So everything I think is laid out in the assignment, but I have an updated assignment that has that family stuff added to it. So make sure you do that part. Once you do the control, it's like, it won't take any time at all. And you can do, well, you can actually do both, right? So you can do it all at once. So I can go to open DNA file. I can get my DNA for the control. And then I can go open DNA, go in, let's say we want to do the Brown family. I can get their DNA. Now I've got all this DNA that I can do together. If I wanted to do PCR, I just get a primer. The primers are going to be the same in each case. Now I select all of them. Run a PCR. Don't forget to run PCR. It may be very easy to forget to do PCR and just run a gel. Now I can select all these guys and let's run a gel. Let's do a quick load gel. So what's it going to look like if I run a gel? There can be any differences. They can be all the same. Let's find out. They're all the same because all we did so far is PCR. So the PCR products will always be the same. And then we would have to digest those with restriction digest and then run them on a gel, and we'd have our 3 controls and our 3 patients. And then you can just compare the patients to the controls and tell what their genotypes are. So you can do it. You can just do it all in one step. You don't have to do the control and then go back and do the family, but you can if you want. Even if you had 2 different gels, that would be okay. Let's see. We can, while we're here, let's click on that. Go over here. And that's the DNA sequence of our PCR product. That's kind of cool. If we wanted to blast that or something, we could do it. All right. I think that's all I wanted to do for today. Yep. Yeah. So just read the assignment and do what it says there. All right. How's the organic going? It's what? Yeah. Lots of information. Well, the make my stepson's doing that now. He's doing that through Michigan State. So I got to listen to a whole organic chemistry lecture on Zoom yesterday. So that was pretty exciting. And I couldn't understand anything the lady was saying. So, anyways, all right. So I think that's it. Unless you got any last questions. All right. So I'm going to sign off. Send me an email if you have any more questions. Adios. See if I can figure out how to get out of this thing.